Okay, I'm sorry, I can't follow these guys. My talk is going to be a lot more boring. I'm sorry. Let me just give a little bit of a disclaimer here. I'm a basic scientist. I'm going to talk about Schwann cell biology and CMT. I'm not an MD. So if we have questions during my talk, luckily we have several MDs who specialize in CMT. They can answer those questions. So I'm going to talk a little bit about some of the basic biology. And I know a lot of you already know a lot of this, so you can go back to check in your email or whatever you want to do. I also don't like to stand out in front of everybody. I like to come down and be part of the audience. Okay, can you all hear me okay? All right, so what's my story? Who am I? Why am I doing this? Well, years ago, actually probably about 25 years ago, I got interested in studying Schwann cells. I just thought they were really cool cells, and I'll tell you a little bit about why that is. They're really interesting. And then as I began studying Schwann cells, my nephew was born, which was great. We were all excited. I don't know why I'm getting emotional about this, but anyway, there was something wrong with him. And the doctors couldn't figure it out for a long time. And eventually they did diagnose him as somebody with Charcot-Marie-Tooth type 2. He really was never able to walk. He's very severely disabled. And he's 23 now. He's struggled with trying to be independent, as you can imagine, as a young man who can't go to the bathroom by himself, can't take a shower by himself. It's really a struggle. And that really sort of gave me a new perspective on Charcot-Marie-Tooth disease. And we had done a little bit on it, from a scientific point of view. Then I really realized all these little things that the patients and their families have to go through. And that really made me want to work a little bit more on really focusing on how to help this disease. Okay, so what is this disease? So you probably have heard it's named after these guys. So this is Jean-Martin Charcot, a French guy. He's actually considered the father of modern neurology. So he discovered and described a lot of diseases, Charcot-Marie-Tooth being one, and multiple sclerosis being another. So he's pretty famous for that. And then his student or assistant, Pierre Marie, was also key in describing that. And at the same time these guys were describing it, this British medical doctor, he was actually a student at the time, described the disease as well, Henry Tooth. And probably some of you in the audience know I have some fish who are named for these guys. Anyway, the disease almost completely only affects the peripheral nervous system, not the central nervous system. So that's good in a lot of ways, because what that means is that you will not have, as a patient, you will not suffer any mental problems. It's all physical. So it's also not great because, like my nephew, then you're kind of trapped and in the body, and yet your mind is completely aware of everything. So what is the peripheral nervous system? Whoops. So the brain and spinal cord are the central nervous system. And then the peripheral nervous system are your sensory neurons. So that's the neurons that give you a sense of touch, pain, heat, cold, things like that. Like you can close your eyes and you know where your limbs are at this point, right? Those are proprioceptors, and they give you that information. That also includes your autonomic nervous system. So like when you're standing in front of a bunch of people talking, your hands are sweaty, your heart's beating a little fast, that's your autonomic nervous system, your sympathetic system. And then sort of kind of part of the peripheral nervous system, but also part of the central nervous system are your motor neurons. So those neurons are actually located in your spinal cord, and they project out into the muscles, and that's where a lot of the problem is, frankly, in CMT. So they're actually in the central nervous system that project out, and the Schwann cells myelinate those. So when I talk about neurons, what am I talking about? So we know nerves. Those are the things that stretch out in your body and give you the feelings. Well, the cell is called a neuron, and it has a cell body here. This is where the DNA gets packaged in the nucleus here. And then it has these really long processes that go out into the different parts of the body, your muscles and so on. And it's wrapped by these cells called Schwann cells, and those form myelin. Now, this picture here isn't really to scale. It depends on the neuron, but the motor neuron that's in my spinal cord and projecting down to my foot so that I can walk around, that neuron stretches a really long way. So if that neuron was the size of a basketball, that nerve fiber called an axon would be 6 miles long. So just think about that for a minute, because what that means is that cell body, this is where all the business is happening here, has to have signaling mechanisms that go back and forth to communicate all that information all the way down to my toe. So there's a lot of things that have to go back and forth along that projection. And then these neurons are wrapped in a group of cells together called the nerve. So this is what the peripheral nerve would be like here. And you can see this is the neuron, and these axons are projecting out of the nerve toward you, and they're wrapped by other cells that form a layer called the endoneurium. And we'll talk a little bit more about this. This is what is going to give you your blood-nerve barrier. So this is one of the challenges for getting drugs into the periphery. And so these nerves then carry the fibers. Like if you have your sensory neurons, they're right next to the spinal cord here, and then they project out to your limbs and give you those feelings of pain, touch, and so on. And they project into the spinal cord so those signals can get up to your brain, or maybe just do a little circuit in the spinal cord there. And then the motor neurons here, like I said, are in the spinal cord, and then they come out to the muscles. And then they have a specialized ending when they get to the muscles. So they form this thing called a neuromuscular junction. And this is another area of challenge for people with CMT, because this is an area where the communication stops. You need to have the signal come down, come down the axon, that big long nerve fiber, and then connect up to the muscle to tell the muscle to move, to contract. And so that's one of the things that starts to break down in CMT. And here's a picture here I took from this paper from this group studying CMT, Michael Serreta. And this is the nerve fiber here in red and coming down to the muscle in green. And these are this specialized little endings here where it communicates with the muscle. And this is a rat that has CMT1A, and you can see that the connection there isn't very good. The nerve fibers are just not connecting very well. And then they gave the rat that drug you might have heard about, PTX-3003, and they could see restoration of that neuromuscular junction. So that drug is actually in clinical trials. It's not great, but it does seem to help. So it's one of the things that is benefiting from the input of people like you and the CMT Research Foundation. So we are making progress here. Now the nerves what they do is they carry electric signals so they can communicate with the muscle. And in order to do that quickly, like if we're walking around, I have to be able to move my muscles very quickly in order not to fall, right? Or if I step on a piece of glass, I want to move my foot really quickly off of that piece of glass. And so that signal has to happen really fast. And so that's where the myelin comes in, because the myelin creates this way of conducting that electric signal so it can go really fast up to my central nervous system, my brain, so that I can respond. But not all the fibers are myelinated. Some of the fibers, you don't need to have it happen real fast. So like if I did step on some glass, you know how like 2 days later you still have pain in your foot? Well, that's because those nerve fibers are continuing to tell you, hey, you have a sore there, you need to take care of it. And so that doesn't need to have a really quick signal, that can be a slower signal. And so those are not myelinated. That's carrying that electric signal a little slower. We'll talk a little bit about some of those nerves in a minute. Okay, so this is just a picture of a nerve. So in this case, those axons, the nerve fibers coming straight out at you. And what you see here are the axons. That's the neuron that we were talking about a minute ago. And then the Schwann cell is wrapping around that neuron and allowing the neuron to signal that electric signal quickly. And these Schwann cells stretch all along the axon for quite a ways. So what you see, like in this picture right here, the Schwann cell is right in the middle. So that's the nucleus where the DNA is, and then it's wrapping that axon right there. And then this one, it's just further along away from the nucleus there. But you also see here, these are those fibers that carry that slow signal, the one that says, oh, you stepped on something 2 days ago and it still hurts. Those are in what they're called a bundle of Rimac, named after another famous guy. And what I want to note here is, you see how there's gray stuff in between little white balls there? That gray stuff is another Schwann cell. And it turns out those Schwann cells are really important for those type of neurons also. So a lot of times you'll hear people talk about Schwann cells as kind of like the coating on a wire. So it allows this electric signal to go down the wire quicker. I don't really like that analogy. I think Schwann cells do a lot more than that. I really think of them more in biology, what you may have heard of called mutualism. So mutualism is two organisms that benefit each other by being together. And so like some classic examples are the honeybee who helps out the flower by pollinating the flower, and at the same time the flower helps the honeybee by giving it nectar to make honey. Or clownfish that hide in a sea anemone, and it gives them protection, but it also protects the sea anemone from its predators. And so they both benefit. So the Schwann cell and the axon, they're like that. They both help each other out a lot. And in fact, if you get rid of the Schwann cells early in development, keep pushing the wrong button here. This was actually done by this group in Germany, Carmen Birkmeyer. They got rid of the Schwann cells, and these sensory neurons in this area called the DRG, dorsal root ganglia. Anyway, these sensory neurons all begin to die off. So the Schwann cells are necessary for those neurons. Just as another example, this group in Washington University at St. Louis, they made a mutation, and this is Jeff Milbrand's group. They made a mutation in the Schwann cell in a gene that affects the energy source in the Schwann cell, the mitochondria it's called. And because the Schwann cell didn't have a lot of energy, the Schwann cell was okay, but then the neurons began to degenerate. We've seen a similar thing. We published a paper couple years ago where we had a mutation in the Schwann cell that messed up the lipid production, so the ability to make fats. And the Schwann cells were okay. In fact, even the myelin looked okay, but we lost 30% of our sensory neurons. So we know there's actually a lot of communication between the Schwann cells and the axons. Another great example is Schwann cells are thought to provide energy for the axons. So like your muscles and different regions in your body store energy as glycogen, right? Starch, basically. You take up glucose and then you store that when it's needed. Well, the neurons don't do that. Neurons don't store glycogen, but Schwann cells do. So there's some evidence that the Schwann cells are providing energy to the neurons in order to fire all the way down the axon. So that big, long thing, well, it's 6 miles, that has to have that electric signal go all the way along there, and it takes energy. So there's this idea that Schwann cells provide that energy. And then most important and exciting, and one of those reasons I think Schwann cells are so cool, Schwann cells actually promote regeneration. So in the central nervous system, that myelin is formed by a different cell called an oligodendrocyte. And that actually blocks regeneration. But in the peripheral nervous system, the Schwann cells actually promote it. So they're helping out there. So when things go wrong, there's a lot of different forms of Charcot-Marie-Tooth disease. This is just one example of a way of looking at this where we have all these different forms. The most common by far is the gene duplication of that myelin protein, PMP22, you've probably heard about. So that accounts for more than half of the patients who have Charcot-Marie-Tooth. A deletion of one of those copies, we each get a copy from our mom and our dad of all of our genes. Deletion of one of those copies gives you a disease called hereditary neuropathy with liability to pressure palsy. It's a big mouthful. We just usually say HNPP. It's easier that way. And then there are all these other types, as we just heard about from Alex and Jamal. There's CMTX, which is a mutation in a connecting protein that connects Schwann cells. And then there's a variety of other types as well. I won't describe them all. But we know there's over 100 different genes that can give you different types of CMT. And there's more being discovered all the time. In fact, about 40% of people with type 2 disease, including my nephew, are undiagnosed. They don't know what the mutation is. So there's still a lot that needs to be done, particularly in the type 2 area. So there are kind of two broad categories when I talk about type 1 and type 2. What does that mean? So type 1 is usually where there's a problem with the Schwann cell. And the way the doctors diagnose it is they can look at what's called nerve conduction velocity. So how fast does that electric signal go down your nerve? And if there's a problem with the myelin, it goes slower. And so they have a cutoff here of a certain amount, how fast it is, 38 meters per second. So that electric signal can go that fast, or if it goes slower, then it's usually type 1. And they can also measure this thing called compound muscle action potential. So we talked about that neuromuscular junction. So how big is that signal when it gets to the muscle? And they can measure that. And so that's type 1. And as I said, that's usually a problem with the Schwann cell where it's not myelinating correctly. And then type 2 is more when the problem is with the neuron. And there, typically the nerve conduction velocity is pretty normal. There is a reduced compound muscle action potential. The signal when it gets to the muscle is smaller. And then there's evidence of axon degeneration. And there's lots of different neurological tests they do as well. And I'm sure the folks here could tell you more about that who are doctors. And then there's the intermediate forms, like we just talked about CMTX, that can be kind of in between and show different properties of both types. And then this is just a picture of some of the different genes. I'm not going to go through them all, but as I said, there are different forms that give you the axon type. And interesting, some of these are problems mostly in the Schwann cell, even though the outcome ends up in the neuron. And so that is another example to me of where that Schwann cell is helping the neuron or hurting the neuron. And the problem is actually in the Schwann cell, though. And then there are types that are primarily demyelinating, like we talked about PMP22 being duplicated or missing, and that gives you the different forms of CMT, like CMT1A or HNPP. And then when you get a diagnosis, and I'm sure many of you had the experience, or at least some of you I know have had the experience where it took a long time to get the diagnosis. It's difficult, and the doctors have a wide range of things they can look for. One of the key things is history. So if you find out, well, grandpa had CMT or some other disease, that is really critical so that they can start to think about what your disease, your child's disease might be. And then they do a variety of different testing. You get the little rubber hammer where they're tapping on your leg or your arm. That deep tendon reflex is what they're looking for. So I'm sure you've experienced a lot of that, or a lot of you have. They also do that nerve conduction velocity thing we were just talking about, like how fast does that electric signal go? And at least my understanding, I actually haven't had that done. Is that, somebody told me that's quite painful. Is that, yeah. And even worse, so like what my nephew had to go through is they actually had to do a nerve biopsy on him. I imagine some of you might have had that too. For a lot of the different conditions, that's not done anymore, as long as they can diagnose it without that. And in fact, for some of the research we're doing, I really wanted to get some fresh nerve tissue from a patient, but the doctors, with one guy in particular, I was collaborating with Jun Lee, he's like, yeah, we can't do that, that's not ethical anymore, because the patient, they don't need to have their nerve biopsied, we know what's wrong with them, what type they have. Anyway, so there's a wide range of things that doctors can use to get the diagnosis correct. And then one thing that's constantly being done is there are people, like I said, who don't have a genetic diagnosis yet, and so they often will take DNA samples and send them to groups like Zuckner, who many of you may have heard of, is doing a lot with looking at the genetic causes of CMT. So when we talk about this gene duplication, what exactly does that mean? Well, as I mentioned, and I'm sure many of you will remember from basic biology, you get one copy of a gene from your mom and one copy from your dad. And what can happen is part of that chromosome can get duplicated. Now, this does actually happen spontaneously in about 5 to 15% of the cases. So not every time somebody has CMT1A is it just because mom or dad has it. Sometimes it spontaneously happens. And it's not one gene that actually gets copied, it's this whole portion of the chromosome. We do know that the problem giving you the disease is that gene, because we can just give a mouse an extra copy of the gene and it shows a lot of the same phenotype, the same properties. And then if you lose that gene or that region, that's what gives you HNPP. So that's the other form of CMT. And then the point mutations there's a lot of them. So this is a little diagram here of what the protein looks like. So this is the cell membrane. Sorry, I'm getting into a little biochemistry here. And then all of these little circles are the amino acids that make up the protein. And all the colors here, like the black and red, those are known mutations. And it depends on where the mutation is as to how severe the disease is. So if you have a mutation right in the middle of this membrane part, it usually makes the protein really unstable and doesn't fold very well. And that gives you a more severe form of the disease. On the other hand, if you have a mutation that's not in the membrane, like this T118M, actually 1% of the people in the world have that mutation. I was working with this friend, Chuck Sanders, who recently published that paper. So it's actually a very common mutation. Now, for most people, I don't know, maybe I have it. For most people, it doesn't cause any problem. But for some reason, some people, it does cause a pathology. You can ask Lily about that if you want to know about that. What exactly is happening when the mutation happens there, we don't really know. But in some people, it gives them a propensity to have a form of the disease. So the symptoms, all of you can tell me a lot more about this, but just so that those of you who wonder what the different symptoms might be, often it's diagnosed early with some difficulty walking. People are just kind of clumsy, they can't really run, or even walk with a normal gait. That's often what raises some awareness to the parents. Also, if you have weakness in the muscles. I remember when Nick, my nephew, was diagnosed, or before he was diagnosed even, we would lift him up in the muscles, which just didn't seem as firm as you would expect in a little baby. And then we have the classic symptoms that a lot of times you hear about, pes cavus, which is just a really arched foot. You also see the hands tend to lose the muscle strength, and so you get that kind of curling up of the hands. The problem is the muscle connection. And so the reason that's happening is because those muscles aren't able to maintain their strength. And so that muscle wasting leads to the weakness, and then that gives you those kind of symptoms. And the same thing is, we often refer to this as inverted champagne bottle legs, where the legs become really thin, particularly distally, and that's because the muscles have become weaker. And then there's a lot of other things that can go along with it. Some of the patients have pain. Hearing loss is something that actually is being researched more now. We didn't realize that this was so much a problem. And it's not just like a general hearing loss. It can be a very specific type that can be harder to find. The audiologists often call it silent hearing loss. One of the things that I found interesting was there was a paper from Mike Schei's group that described small fiber weaknesses or problems. They wanted to see, okay, I had mentioned the sympathetic neurons, the ones that give you that fight or flight response, make your heart beat fast. Well, those are not myelinated. There are Schwann cells or glial cells associated with those neurons, but they're mostly not affected. So the good news is, like your heart, you're good, no problem there. But some of those fibers, for some reason, are. So a lot of patients report what's called hypohidrosis. In other words, they don't sweat much. Not a bad thing, I guess, but I just thought that was really interesting. So what does the nerve look like in the patients? Again, this is where the fibers are coming out at you, and we do a little cross-section, and then this is actually done by an electron microscope, so it's very high resolution. You can really magnify it a lot. And so this is the healthy nerve here. You see the axon and the Schwann cell wrapping around it. And then these are those fibers that are not myelinated but connected to Schwann cells. And then this is what it looks like in some of the different conditions of CMT, where you get this decompaction. They call them onion bulbs, where the myelin's not compacted correctly. And that's thought to be that the myelin is not forming correct, and another Schwann cell comes on top of it and tries to form, and so you get this kind of big wrapping but not tight wrapping. And then similarly, they get these things called tomacula, which kind of looks like a sausage. And that's where the myelin has become really thick and it's not forming properly. But eventually, the main problem is loss of the axon. So as I said, those Schwann cells are really important for the axons, the nerve fibers. And so if the Schwann cells aren't functioning correctly, then the axons start to degenerate. And that's where the problem comes in, because they can't communicate to the muscle. And then you get muscle weakness. The axons can't carry that signal to the central nervous system of touch and heat and proprioception, you know, that thing where you can close your eyes and you know where your limbs are. So when that happens, that's where you really get the symptoms. So ultimately, if we can do something to fix the axon, that might be pretty generalizable in helping patients. So the good news, though, as I mentioned before, is that the peripheral nervous system actually regenerates. And the Schwann cells are really key to this. That was one of the things I thought were so cool. So you can take a nerve and you can cut it, completely cut the nerve, the axon here. And then those Schwann cells that were forming myelin, they de-differentiate. They go back to like a state where they're like a precursor. And then as a result of that, they'll start eating some of that debris that's around. So they act almost like a macrophage, an immune cell. They start eating that stuff. And then they express proteins that help the axon regenerate. So those Schwann cells are necessary for that axon to regrow. And it will regrow, and it goes back, and everything goes back pretty much to normal. Now, it's not 100% back to normal, but definitely enough to have function. So where are we with treatments right now? Well, unfortunately, as has already been mentioned, we don't have a lot of great treatments. Mostly what is done is help you deal with the situation. So you can have orthotics. A lot of people will wear those to help them be stabilized and walk. A lot of people end up having surgery, like you've heard about, which is great. I mean, I remember seeing a little video of a little girl who clearly had some severe problems, was walking with ankles turned, and then after surgery was doing gymnastics. It blew my mind. But it is a slowly progressive disease, so that it continues to get worse, unfortunately. So what can we do to treat it? Well, there's a lot of research being done, including some of the stuff in our lab, where we look at different models so we can test different therapies. So we can culture those Schwann cells with neurons, and they'll form myelin. This is some myelin here. You see these little white lines. That's myelin forming. Lots of questions there. You can't see, but there's actually millions of Schwann cells in that culture, and only some form myelin. Why? I mean, I've talked to a couple people about this. We don't know. Why don't they all form myelin? There's also lots of different mouse models. Mice are a great system in order to study some of the diseases. They're mammals. They form myelin. They're not always great. We've actually cured cancer many times in mice, but it hasn't translated well to humans. So that is a limitation. And one of the things that's being done now to address that aspect is we are also using human cells, where we can take a patient's cells, and probably some of you have had this done, where they take some skin cells and they can convert those to Schwann cells and neurons. This is a really cool technology. The Nobel Prize was won for discovering how to do this. And then you can get myelin to form in a culture from humans. Unfortunately, the efficiency in the human is even worse than in a mouse. So we don't know how to get that myelin to form very effectively. I'm not going to go through all the strategies because I just got told I have 2 minutes left. There's lots of ways we can approach this problem, though, and people are thinking about it. Everything from how to manipulate the DNA to how to look at the protein folding, the problems when it doesn't fold correctly. We can target some of these things. We can target the axon to try and restore the axon function. As I said, I'm not going to have time to go through all these different details, but there's lots of approaches that are being taken in order to cure the disease. I do want to just briefly mention one because it's such a fun story. So the Stefan Zuckner, the guy that does a lot of the genetic stuff, he had a colleague who was working with him, Andre Cortese, and they were studying the SORD mutation. Some of you may have heard this story. Sorry, I have to tell you again. But they were looking at a type of CMT, and when they do the sequencing, it'll all look fine of different genes. And they were looking for something that was wrong. And what was causing the problem is there's what we call pseudogenes. So we all have copies of some of the genes that we need that are either not actually made or there's like only a part of that gene. So we have these pseudogenes. And so in this case, the sequencing kept coming back looking fine because the pseudogene had that region just fine. There was no problem. But then when they looked a little more carefully, they realized the gene that actually gets made into RNA and protein, that was messed up. And so once they discovered that, they said, oh, well, it's actually a mutation in this thing called sorbitol dehydrogenase. And what that is it's a protein that's important in converting sugar to fructose. So these are all different kinds of sugars. So we all need glucose. You take up different things in your diet and it gets converted to glucose. We need that energy, and it gets made in some cases to sorbitol and then to fructose. And sorbitol, that's the problem. That's nasty stuff. Too much sorbitol is really bad for you. And it turns out the same thing happens in diabetes. And it's not surprising, right? If you have hyperglycemia, where you have way too much glucose, then what the real problem is, you build up a lot of this sorbitol, and that causes a lot of problems. There's other things wrong too, but sorbitol is bad. Okay, so they discovered this problem in sorbitol dehydrogenase, this protein here. So if that's not working correctly, what if we could just shut off this pathway? Well, they already are doing that for diabetes. So there are current drugs that target this enzyme, this protein here. So it stops that pathway. So the amazing thing is, in just a few years, they went from diagnosing what was wrong with those patients to actually having a treatment. So there are clinical trials now using some of those diabetes drugs for that SORD mutation. Because we're out of time here, I'm just going to mention a few other approaches very briefly. Some of you may have heard of ASOs or antisense therapy. What's being done there is your DNA gets made into RNA, and then that's made into protein. So if we could target the RNA so it gets degraded, then we could get rid of the problem RNA. Like if you have too much PMP22, we can target the RNA with what are called antisense. There's a sense strand and an antisense strand. And when you get double-stranded RNA, the cell says, oh, that doesn't look right because we don't normally have that. So a lot of times it's a virus. And so there's a system set up to degrade it. So if we artificially make double-stranded RNA, then your cells will degrade it. And then CRISPR is a new technology. Sorry, I'm going to go over a little bit because I have to tell you, as a basic scientist, I want to tell you this really cool thing. Why do we know about CRISPR? Right? You've heard a lot about this. It can go in and it can edit your DNA. It can change the DNA. It can fix the problem. If you have a mutation in the gene, we can go in and fix it. You know why we know about that? because scientists were saying, I wonder how bacteria fight off viruses. Yeah, bacteria get infected by viruses, and they have a way of fighting it. They can use this technology we call CRISPR, where that's how they figured out how we can take advantage of what bacteria do and now treat humans. So that's a great example in my mind of how just understanding the basic science can lead to dramatic changes in therapies. And so now there's different approaches being made to try and get this gene editing system into the nerve and the Schwann cells. And as I mentioned, one of the great problem, the blood-nerve barrier. So somehow you have to get the CRISPR or the antisense or the small molecule across that barrier so we can get to the Schwann cell or the axon. And that's the problem. I'm out of time now, so I'm not going to go through biomarkers. There's a lot of excitement about biomarkers. And natural history is really important. I will mention a little bit about that. Every patient needs to make sure they go and get a natural history done, period. And the reason is, for really common diseases, we have a whole bunch of people, and we can say, okay, we have these markers, and we can figure out, okay, with all these people, we can do a trial. We can take a placebo, we give 1,000 people a placebo, we give 1,000 people the real drug, and see what happens. You can't do that if it's really rare disease. You might only have like 50 people. So what are you going to do? Well, what we have are these natural history where they look over time what happens to the patient. So if we say, okay, here's where the patient was at 3, here's where the patient is at 7 or whatever, then they have this natural history. And so by having that, giving them a drug, then we can say, did they get any better? Did they stay flat? What happened with that drug? So that can be like on an individual basis. You hear about personalized medicine. This is one example. But the disease is correctable. So if we fix it, you'll get better. And we heard about that yesterday from one of the researchers working on this disease called giant axon neuropathy. And they actually, in adults, got better. And this is just an example here where they used a mouse that overexpressed PMP22. It had CMT1A. That mouse was just like many patients. But then they shut off that extra copy in the adult. And the nerve got better. And so that the mice were pretty much back to the wild type. So I'll end there. Sorry I went over. But I think that there's a lot of excitement in CMT right now because there are some real therapies being developed, and we can actually restore function even in patients who are further along. So the impossible becomes possible. Thanks.
Bruce, thank you so much.